Okay, so it is cell communication day 4. So we started on Monday talking about like what types of signals cells can send. We talked on Tuesday about different types of receptors, and we had 3 specific receptors. Yesterday, we talked about signal transduction pathways, how we get from receptors to the response. And today, we're going to actually talk about how we manage that response. And there are 4 big kind of components to that we're going to talk about. So, if my stuff would work, maybe. There we are. All right. What happens after transduction? That's what today is all about. So, typically, cell signaling leads to the regulation or transcription, or regulation of transcription or cytoplasmic activities. So, something goes on in the cytoplasm, or we get genes transcribed so we can later translate them into proteins. A lot of them ultimately regulate protein synthesis. Um, I don't think we've gone super in detail about protein synthesis yet this year, but it is essentially that whole transcription translation going from DNA to mRNA, mRNA to proteins, using these special codon charts and all that fun stuff. If that's ringing any bells. So, what signal transduction does a lot of the time is regulate what mRNAs are transcribed. So, what genes we're actually going to make copies of, so we can make proteins. And sometimes they even regulate protein activities like ion channels and metabolism. So, all of the stuff we learned last unit with cellular energetics, that can be regulated through these, by these signal transduction pathways. So those are our big ones for this unit. Really, the big things that signal transduction pathways can do: regulate transcription, therefore controlling protein synthesis, or regulate protein activity. Is what I'm looking for. Regulate protein activity, and therefore leading to things like metabolism. That is both, anabolism and metabolism. So after transduction, when we say like cellular response, these are typically those responses. So making new genes, turning off genes, making new proteins, turning on and off proteins. That's the point of all of this stuff. So whenever we talk about protein synthesis and stuff like that, what we're going to do is we're going to kind of review back to this and be like, okay, hey, remember when we said when we talk about signal transduction pathways, that's how this RNA polymerase knows when to start transcription. So it's going to be one of our big links between units. But responses, these cell responses, is a lot more than just like turning things on and off, right? I mentioned turning genes on, turning genes off, turning proteins on, turning proteins off. But it's actually, as in life, more complicated than that. There are 4 big aspects of how we regulate the response through these signal transduction pathways. The first is amplification. The second is specificity of signaling. The third is the efficiency of the response. And the fourth is the termination of the signal. Because once a signal gets sent, we also have to be able to, turn it off. It's not like a cell receives one signal and then they constantly carry out that cell response for the rest of their little cell life. So we have to be able to shut it off. We can regulate how big a response is by regulating the efficiency of it. We can make a response stronger through amplification, and we can have different responses to the same signal through the specificity of signaling. Remember, enzymes are proteins. In proteins, shape determines function, which means that different proteins, different shapes, different functions. So these are our four big ideas. And that, if you got the objective at the beginning of the slides, um, knowing these four things and just kind of being able to describe what each one of them means to cell signaling is the objective for the day. Making sure we know that these four ways are how we can regulate a cellular response. So we're going to start with signal amplification. So when we say the word cascade, like phosphorylation cascade, right, it kind of has a gravitas to the word, right? Cascade isn't just like this happens and this happens and this happens. It feels like it gets bigger as it goes on. And that is, that's exactly what happens. We have this signal amplification. So each step, the number of activated products is much greater than the last step. It's like the pyramid scheme, the MLM of cell signaling, right? One person at the top, one big signaling molecule activates another. That one activates 3. That one activates 5. That one, each one of those activates 2. So when we have one signaling molecule up here with number 1, we can actually see how we can get from that one signal to like, what is that, 2, 4, 6, 8, 10, 12, 14, 16, 18 activated proteins that can go throughout the cell and do whatever they need to do. All because signaling molecule 1 turned on a response through 2 into 3. Right, number 1 is our signaling molecule, that's our ligand. Number 2 is our receptor. The receptor activates this protein here. This protein, number 3, activates 3 intermediates in that cascade, in that transduction pathway. Each one of these 3 activates another 3, and each one of these activates 2. So our number of activated proteins gets bigger and bigger and bigger with each step. So the signal gets stronger and stronger and stronger as it moves down this pathway. So if a cell response needs just like a small signal to get started, maybe these won't be as dramatic. Maybe we'll see like 1 or 2 steps. But if we need a fast signal, super, super fast, and to spread through the whole cell, we can have like 6, 7 of these steps with way more products. So signal amplification is one way we can regulate cell responses. Specificity, that's a fun word. Specificity of signaling can also help coordinate responses. So different cells have different collections of proteins, right? The proteins that are floating around in your heart cells are way different than the proteins floating around in your liver cells. So the response can depend on the receptor a cell has, right? If your body is releasing a hormone that is meant for your muscle cells, right? We need to get a signal to your muscle cells, but we don't want that signal to activate something in your lung cells or your skin cells. We make sure that the hormone, that release, is only being able to be received, so only the muscle cells have the receptor for the protein that we are sending out. Another cool way that this works is that cells can have the same receptor. So like a lung cell and a skin cell can have the same receptor, so they respond to the same molecule. But the proteins participating in the cascade, in our phosphorylation cascade, can lead to different responses. So if you think about like the process of the human body going through puberty, right? We release a lot of hormones, but they are a lot of the same hormone, but your body has different responses to it all the way through, right? Um, you've got your sweat glands responding to it in one way. You've got your hair follicles responding to it in another. You've got your stomach cells and nerve cells responding to it in different ways. So they all have receptors to to this hormone, this signal that's being sent out en masse. But we have different responses, and that is because there are different proteins inside the cell that will participate in that cascade. So, different proteins mean we can use the same signaling molecule but get different responses, right? A very cool way that our bodies have kind of increased efficiency. I say our bodies, like all multicellular organisms have very similar processes. So that is our specificity of cell signaling. Efficiency was our third one, right? Being able to regulate how efficient a phosphorylation cascade is. So if we're like looking at a cell system, right? A lot of the time, stuff's just kind of free floating in the cytoplasm. And it's not like our proteins have little GPSs on them to help find other proteins. They don't have Find My Friends, right? So a lot of times it's just like random chance and diffusion that two molecules that need to participate in a reaction will actually meet up with one another. But cells are smart, cells are clever, and they found a way around this, and that way is scaffolding proteins. Scaffolding proteins will increase the efficiency of the signal by holding stuff in place, right? If you're building a house, right, it's going to be a lot more efficient process if you've got scaffolding to help get up to where you need to go. So it's a lot more efficient process with scaffolding proteins holding things where they need to go. All a scaffolding protein is a large protein to which other proteins are attached. So if we see this little diagram right here, we've got our signaling molecule and our receptor. This can be any one of our three receptor types, just like generic receptor. Our scaffolding protein, this one is actually going to bind directly to the receptor and then activate three different protein kinases, which can then go off and do three different processes. Or if they're the same protein kinase, amplify that signal, just like our signal amplification thing. Right? So the scaffolding protein can hold a bunch of stuff together. That way, we get stuff done more efficiently. Right? That's what our scaffolding proteins do. They hold stuff together to make the process go more smoothly. All right. So, some specific scaffolding, a lot of them, specifically the ones in synapses, so the little spaces between nerves or between nerves and muscles, will actually hold networks together, like holding the steps together. So, in this one right here, we've got the difference between free diffusion and a scaffolding protein. So in our free diffusion, our stimulus is going to activate one protein, and it has to go off and find the second protein, and it'll activate that one. The second protein will have to go off and find the third protein, and this takes some time. If we use a scaffold, we can hold those three proteins close together so they don't have to go off and find one another. So we've got our same process here, 1 to 2 to 3, but they're held super tightly together. That way we can get stuff done. Some cases, our scaffolding proteins will actively like participate. Other times, they're just there to like hold stuff together. So sometimes we can have them that will actually like turn on this first one. So like the stimulus comes in over here, and then it will activate the first, which activates the second, which activates the third. Or the stimulus can just come directly into our first one. It just depends on which system we're looking at. Almost there. So, termination of the signal. If we want a signal to end so we can receive another signal, that kind of depends on reversibility. Our cell responses only occur when signals rise above a certain threshold. But in order to receive another signal, right, we have to return to inactivity. Like, I mean, the metaphor doesn't really hold now that we've got like call waiting, but it's like if you're trying to call somebody but they're on the phone with somebody else, right? They're not going to get that kind of message. They're not going to be able to answer your call if they're on a call with somebody else, right? Our cells aren't going to be able to receive a signal if the first signal isn't shut off. So those signals don't stack. In order for our cell to be available to receive a new signal, they have to return to inactivity. They have to return to that kind of baseline, no response, all that fun stuff. Questions about any of this? Amplification, termination, efficiency, specificity? We feel very low energy today. All right. So, this next bit, you do not have to write down, but we're going to do an activity that requires some background knowledge. So, I want to give you some background info so we can take that into our activity. So, we're going to do a quick case study in cell communication. So, we're going to look in depth at an example of how cells communicate to one another. And we're going to look at that through apoptosis. Have we talked about apoptosis in this class yet? No. Okay. So, apoptosis is programmed cell death, essentially. And it is a super good example about how multiple signals can be kind of used together to get a cell response. So, apoptosis, like I said, is programmed cell death. The signal to commence apoptosis can come from multiple places. It can come from outside a cell, where another cell says, hey, buddy, you're not doing too good. We need to protect this organism. I'm going to need you to be no more. Or it can come from inside the cell when something goes wrong and they have to kind of hit the self-destruct button to protect the rest of the group. The goal is protection. If a cell has a really bad mutation or if something harmful is in the cell, we have to target that cell for death, right? If this doesn't happen, if apoptosis doesn't happen, that's how tumors and cancers form. And we're going to talk about that next week when we talk about the cell cycle. So during apoptosis, what happens is that cellular agents inside chop up the DNA, because if that DNA has a mutation, we don't want that out there. We don't want to risk anything picking that up. So we just kind of munch up the DNA and fragment all of our organelles. And we take everything and we kind of package it up into these little blobs that are very scientifically called blebs. Right? The process of this is actually called blebbing. I do not know who made it up, but I think it's hilarious. And so all of those little blebs are vesicles, and we have special cells that are really good at taking in those blebs, digesting them, and then using them for spare parts, essentially. So that's apoptosis. What we're going to do is I'm going to have you partner up. There will be one group of 3 because, odd numbers. And I have quick, small little examples of how apoptosis is signaled in C. elegans, which is a little worm that is very, very popular in genetics and development research, and then animals and mammals. So what we're going to do is you're going to have 10, 15 minutes to quickly read through this and get yourself a good description of what's going on. Whether that be drawing pictures on the back of your page or jotting down bullet points, you're going to have so much time. I'll figure it out when I do some math. You're going to have some time to read through and get an idea of what is going on in your example. Your partner is going to have the other one. So one of you will have the C. elegans page, and one of you will have the humans and other mammals page. You're going to become an instant expert in your page, right? Then we will have some time. We'll take turns. One of you will explain your apoptosis pathway to the other, and then we'll switch. Because I found one of the best ways to learn is by explaining it to other people. If you can put it into words, it makes you think about it. So, what I want you to do is I want you to partner up around the room. Find a place, and I will come to you.